Good afternoon, everyone, and welcome to the third HBP Tea Slides. For the new ones, the education program of the Human Brain Project, together with the student representatives and the ambassadors, designed this format so that young researchers and students in the HBP could present their work and share ideas and contents with other students working in the same field. Maybe starting new collaborations, networking with other scientists, getting to know what other groups are working on, and maybe starting to use new tools. So we hope you will enjoy these tea slides, and from that start new ideas, new networks with the other students. Hoping that we will have chances to meet in person at the next HBP education program events. The tea slides is organized in 2 talks of about 10 minutes. If during the session you experience any technical problem that you cannot solve, we already apologize for that. And you can send an email to the education program that you can find the email in the slides here, and we will try to fix for the next sessions. After each talk, we will have a Q&A session. You can ask your questions in the Q&A panel in the application, and the speakers will answer after their session or in the final discussion, and they will also be available to answer by email in the next days. Or after each talk, you can also directly do the question to the speakers, raising your hand with the button you find in the application, and you could directly ask them. The material of the event will be available in the e-library of the education program, so it will be online in the next days. You will find the recording of the session and also the slides. And we can start with the first speaker, who is Juan José García Cantero from the Universidad Rey Juan Carlos in Madrid, Spain. He is a PhD student working at the Computer Engineering Algorithms Computer Graphics Department. And today he will talk about generation and representation of brain anatomical structures. And in his talk entitled Techniques for Visualization and Improvement of Brain Structure Description.
Okay, thank you for your presentation, Alice. Okay. Then, as Sally said, I'm going to present the techniques for the visualization and improvement of brain structures descriptions. First of all, I would like to comment that I recently accomplished my PhD at the GMRE group of the Rey Juan Carlos University. My thesis is related to the scientific visualization field, specifically focused on the efficient representation of brain structures and on the improvement of their computational descriptions. Following in this presentation, I'm going to show you 2 applications obtained as results of my thesis. Neurotestmes focuses on the efficient and accurate representation of neurons to allow the analysis and visual exploration of scenarios composed by large amount of these neurons. You can see the user slides. In this case, the representation of neurons membrane is based on the generation of neuromorphology tracing. These tracings have an incomplete description of the neuron anatomy. For that, the generated cosmos is subdivided during the visualization using the hardware tesselation provided by the GPU. This subdivision can be applied with an adaptive criterion depending on the camera distance that allows the visualization of the neuron membrane with interactive frame rates. In the a kind of this description for neurons where the neurites are described as a combination of points, and the soma is described as a contour. Here you can see an example of the cos meshes that we have generated for each neuron. This mesh is composed of that will be subdivided by the hardware tessellation. One special part of this coarse mesh generation process is the reconstruction of the soma surface starting from the incomplete information available in the morphology. In the figure, we can see the contour of the soma. That is the only information that we have to reconstruct the soma surface. This information, as I said, is described only with a contour, and in some cases we only have the center and the radius of the soma, not even the contour. Consequently, to obtain the soma surface, we propose the reconstruction, a reconstruction where an initial sphere is deformed in the direction of the dendrites, as you can see in the images. This deformation is performed simulation of an elastic material using the finite element method. In the figures, you can see the evolution of this simulation and how the surface is tend to the starting of the neurites. This image shows the kind of representation obtained as a result of the refinement process for the whole neuron in this case. The left image shows the application of an homogeneous criterion that gives an reconstruction or a representation of the neuron that have the same level of detail in the whole space. In the case of the right image, this image shows the application of an adaptive criterion on the camera distance, where the neuron areas that are closer to the camera are represented with more geometry. As you can see, for example, here this part of the soma have a lot of geometry, and the the parts of the neuron that are far from the camera are represented with very little geometry. In this way, we can accurately represent the neurons while keeping the interactive frame rates for scenarios composed by large amount of neurons. Okay, the second application that I'm going to present provides several tools to the correction and improve neuronal tracing. Among these tools are, for example, the simultaneous visualization of the tracing and the membrane reconstruction tracing. In the image, you can see an example of this of one situation given in this application. This window that you're showing side by side, we have the original tracing, and in the modified tracing, applying the tools that I'm going to present. First off, this tool is the manual selection and edition of the neuromorphology tracing points that compose the anatomy of the neuron in the 3D space. You can select tracing points and displace them or rotate them or wherever. Even you can change the radius. Other tool that this application allows to do for you is the automatic detection and correction of several errors identified as frequent in the tracings. You can select between different errors and also select the action that you wanna do to fix this error. For example, in this example, we wanna check. We want to apply the correction for duplicated nodes in the tracing. We select the test method for duplicated the action that we're going to apply to fix this error. In this case, we're going to select the console out. This means that we're going to get the tracing node there are duplicated in the tracing. Here we have the example and the exit of this example. We have here two buttons with the identifiers of the nodes that are duplicated in the morphology. Then we can push this button and the application gonna give us the selected node that is duplicated. We can manually edit this node, or for example, we can apply a straight correction that is to delete this node straightly. This is only one example of the kind of correction that we can apply with these methods. This implement their own correction methods in Python code and apply them straightly on the application. Here you can see other examples. The zeta coordinates of all the morphology tracing points is set to 0. As a result of the application of this correction method, the original tracing, we get this modified tracing that we can see where all the tracing points are coplanar. The application also allows to export the modified tracing and the reconstructed message generated from these modified tracings. Here you have some interesting links in case you want to go in deep with some of the presented applications. Thank you so much. If you have any question, I'm here to ask you.
Thanks a lot, Juan, for this very nice presentation and overview on these very interesting visualization tools. So let's see if there are any questions. Yeah, from Patriots. So
I can see the chat. If you can tell me.
Yes.
Can you hear me?
Yeah.
Hello. Thank you for the presentation, Juan Jose. I had a few questions, but I'll just stick to one for now and maybe ask some others later if some other people have questions. Can the tools that you've created, that you've developed, be used to generate sort of connectivity scaffold? Can you take something like the shape of individual neurons, say pyramidal cells or Purkinje cells, place them in a volume, and then somehow decide which neurons are connected to which?
Not this application. In the group, we have several applications that trade also with connectivity problems and with the conformation of connectivity between circuit of neurons. But this application is more focused on both are also connected with other applications developed in the GMRB that gives you the chance to modify the connectivity and all of this stuff, and you can visualize which part of the morphology you are connecting. Okay, this you can do this, but not working straightly with the connectivity.
Thank you.
You're welcome.
Okay, thanks a lot for your answer. We have another question in the Q&A. So what types of experimental data can you import as base in your tools? Is there any specific format?
Yeah. Any more? Or...
No, yes.
Okay. Okay, sorry. Yeah, we are working mainly with the SWC format, that is the classical morphology format for neuron, that it's more accepted in the community. But we have also blue configs, for example, for large circuits. And in the case of individual morphologies, we have also worked with the H5 formats, but in very concrete situations. But we can include more morphology loading, and also the ASC format we have for individual morphologies. Yes, that's it.
Okay, thanks a lot for your answer. And so if there will be more questions, we can ask later in the discussion at the end. And now we could move to the...
Just let me point one thing, that is you have the application is in this web page, and it's really compiled. You can just download it and use both applications, NeuroTestMesh and NeuroEditor. You are interested into test them.
Okay, great. Thanks a lot.
Thank you.
Okay, and also you can contact directly Juan Jose in the next days by email. He will be available if you have other questions or need more links and documentation and information. And so let's move to the second speaker, Hari Kalidindi from the Scuola Superiore Sant'Anna in Pisa, Italy. He's a PhD student, almost finishing his PhD at the Biorobotics Institute, working in the field of biorobotics and computational motor control. And he will talk about goal-driven neural network models of biological movement control. Thanks, Hari.
Thanks, Alice, and thanks everyone for attending the talk. So today, in this talk, I'll demonstrate the utility of neural network models that are optimized on realistic task simulations, their utility to understand how brain might be performing similar mechanisms. So to start, we humans and animals can learn effortlessly perform complex motor skills. And decades of control systems research basically meant that we can also make machines that display sophisticated skills. But in order to appreciate the true complexity of brain mechanisms, we have to see what the brain does in case of failures. In many cases, in spite of malfunction in several brain regions and even the body, the brain manages to avoid any catastrophic failures. And it is interesting to basically see the brain processes which are enabling this kind of flexibility in our movement control. But the only problem is, even for the simplest of the movements, multiple brain regions coordinate basically to produce movements. Briefly, millions of neurons control hundreds of muscles, causing a problem of redundancy. And basically what it means is the same kind of movement can be produced by many different types of neural activations. So if we have some neural recordings, how do we interpret why do we see this particular kind of a neural response during the movement? So the underlying assumption for my work was that systems are better understood if we link them to behavioral goals or needs of an organism. To be clear, as an example, in one of the golden paper wasp species, a good facial recognition capabilities were observed. But in a very closely related wasp species, these facial recognition capabilities were absent. So on the surface level, both these observations might seem strange. But if we look at their social needs of each of the species with the high facial recognition abilities have complex social interactions and multiple queens, and facial recognition might help in this setting, while the other species doesn't have such a complex social interactions and have solitary nesting patterns. So while not mandatory, the needs or goals of a biological organism basically can impose certain constraints whereby we can interpret the underlying processes. So the aim is to basically follow similar philosophy in characterizing the motor circuits involved in goal-driven movements. And we conducted 2 separate studies on eye and limb movements, but in this talk, I'll present the results from eye movement adaptation. So adaptation is necessary to adjust motor commands in basically to deal with different environmental or body variables. And in the study, we basically describe how the cerebellum might be helping in this movement adaptation. So to basically study the cerebellar processes during adaptation, we choose a particular class of eye movements called saccades. Saccades are these fast and rapid eye movements that we make to basically point objects in the, point interesting locations in the visual scene to make a mental map of them, or to basically track objects that move in the scene. And typically in this photo, you can see the typical saccadic eye movements that we perform when we look at a face. And also saccades involve multiple brain regions to coordinate with each other to basically process the image and segment it and choose appropriate targets in the visual scene, and basically to move towards that target. But what we are interested in is basically the subcortical structure that is involved in generating the movements. And since the main objective of saccades is to gather as much information from the visual scene as possible in a quick manner, the movements are very fast, reaching up to 1,000 degrees per second. And the movements, they basically last between 20 to 60 milliseconds in case of monkeys, and a little bit higher in humans. So in spite of, since the movement duration is very less, sensory information processing is not very useful to help control the movement. So in spite of not using the sensory processing, the movements are pretty accurate. And how is this possible? So cerebellum is hypothesized to compensate for the lack of this sensory feedback, because we observe that damages of cerebellum leads to inaccurate as well as slow movements. And how is the cerebellum able to achieve this? A hypothesis is that cerebellum continuously monitors the errors between desired and produced movements, and basically uses those errors to adjust the synaptic strengths within the cerebellar cortex, and basically with an objective to reduce the movement errors. So if we consider the generation, how saccades are generated, the main structures are superior colliculus and brainstem. Okay. So superior colliculus has a one-to-one mapping from the basically the visual scene, and let's say a single point on superior colliculus corresponds to initial location, and another corresponds to the target location. So this difference is propagated to brainstem in the form of excitation, and it ultimately leads to an appropriate muscle command. And another feature of saccadic movements is we will be able to find this error only after the end of the movement, since the movements are very fast. And there is another indirect pathway through the cerebellum. And since cerebellum has a capacity to adapt its motor command, this might be helping in accurately controlling the saccadic movement. The only problem is if we use this visual error, the end visual error, to modify the cerebellar synapses, since the visual error at the end instance does not contain any temporal information regarding the movement, it is not useful for compensating the whole sensory feedback during the entire movement. So instead of exploring which signals are present in the biological circuit to basically carry out this task, we directly ask what is the goal of a saccadic movement. And we formulate the goal of this movement in terms of a movement costs. And then we find plasticity rules that can optimize these costs. And these plasticity rules must be biologically plausible. So the goal of the movement, if we write it in terms of a cost function, the first term would be end visual error. So the higher is the movement error, the more is the penalty associated with the movement. And the higher is the time, the duration of the movement, the more is the penalty. And since the accurate information of time is not directly available, we assume that this time estimation must be happening internally within the cerebellar synapses. And we basically hypothesize that this must be happening at the cerebellar outputs, which is basically the Purkinje cell activity. And we did several assumptions in these cost functions, and ultimately we ended up having these plasticity rules that this kind of a neural circuit should have in order to optimize the movement cost. So one of the plasticity mechanism is an error-dependent plasticity, which is very well known in previous studies on how cerebellum adjusts the movement errors. And the other plasticity mechanism we came up with is the error-independent plasticity that doesn't depend on the error, and it occurs during the movement, in contrast to the error-dependent plasticity that occurs after the movement. While error-independent plasticity is well known in cerebellum, but traditionally it is assumed to basically maintain the information content or the homeostasis of the cerebellar circuit. But here we found that this is actually responsible for modulating the movement also. So basically we took all this model and performed realistic simulation on a simulated monkey oculomotor dynamics. And we found that this kind of a model replicates the basically the movement characteristics of monkey eye movements, where the peak speeds reach until 1000 degrees per second, where the amplitude of the movement is 30 degrees, and the movements range roughly in 20 to 60 milliseconds. Now, since we replicated the behavior, we asked if this model cerebellum has anything to do with realistic cerebellum. So recent findings, I'm sorry for not giving a reference here, I'll provide it again. So these are the recordings from recent paper on in vivo recordings from monkeys performing saccades from Shadmaher's group. And basically they saw that the output of the cerebellum, which is the Purkinje cell population, the responses, they correspond to the saccadic kinematic metrics, such as the amplitude and the speed. And similar recordings were similar observations where all can be made in our model also. And this is possible only if we consider both the plasticity mechanisms that we derive. Only if we use just the error-dependent plasticity, we will not be able to replicate the same results, indicating that the error-independent mechanisms are also important for movement. And then once we simulated the biological, neural responses, we tried to apply the same model on a humanoid robot to see if it is applicable for any robot adaptation task. And we saw similar results in robots also, where in the first plot, we'll see that there is an active decrease in the flight time. Basically, the movements become faster and faster as we keep on training the robot, while also the movements becomes more accurate. And in the third and fourth plot, you will see basically we choose random test targets on the camera frame of the robot, and the robot has to move towards those targets, and it is successfully able to move after the training on cerebellum. And now, basically, since it is also working on humanoid, we increased the complexity of the robot. Basically, we used a flexible robot and also replaced the simple brainstem circuit with different kind of basic controllers, like 3 different control architectures called open loop control and closed loop control, which basically continuously monitors the robot state. And the third is basically our adaptive cerebellum control. Now we tested all these architectures on while a soft robot is required to move in a circular trajectory, a target, basically a target tracking task. While we, we will systematically disturb the dynamics of the body. Basically, we'll remove actuations from some module in some trials. We'll completely remove the limbs in some trials and see which of the controllers performs better in these simulations. So these are the simulations. So this is the base task. And cerebellum network indicates basically the network with cerebellum. So in this task, we completely removed actuation from the top module. And the cerebellum network will be able to accurately adjust for the inaccuracies. And here we remove the actuation from bottom module. And in the last task, we completely remove the upper module. So it is very difficult for the robot to adjust for this kind of perturbation normally. While the cerebellum network still tries to do as best as it can. So yeah, in summary, basically we observe that distinct plasticity mechanisms in cerebellum regulate different aspects of movement optimality, which is basically accuracy and peak speeds. And cerebellum compensates for the lack of sensory feedback by adjusting the feedback gains of the control loop. And robot demonstrations show the robustness of cerebellar learning loops.
Thanks a lot, Hari, for your presentation. And we can start first with some questions directly to you, and then we can also do a brief discussion for you both.
Yeah.
First question to Hari is from Mohamed Khalili. Is it possible to import central pattern generator to your work to aim improve quality?
Central pattern generator. I would assume that you mean to import a central pattern generator in place of the cerebellar circuitry, which is actually possible. So yeah, that is possible in the sense. So if you remove or replace this part of the circuitry with a central pattern generator, we should be able to achieve similar things because we didn't impose the constraints on what the cerebellum architecture must be. So it is possible.
And you expect this to impact more in their outcome of learning at the behavioral level, but to lose in biological plausibility, or also to gain in biological plausibility?
Okay. For this, yes, it is actually not yet known whether the cerebellum during saccades, it takes an open loop command from superior colliculus, it continuously monitors the afferent, input from the brainstem. So right now, actually, both solutions are possible without really affecting the behavioral level.
Okay, yeah. Yeah, thanks. So other questions? One is from Sandra Diaz. Which simulation platform do you use for the brain model?
So the code is basically written in MATLAB. We basically wrote the dynamics of the robot and all the neural networks in MATLAB. But recently we switched to PyTorch, which is a deep learning framework that basically automatically generates the gradients of a robot dynamic. So that is much more helpful in performing these kind of simulations. So I would prefer to use PyTorch in these cases.
Okay, thanks. We have other question from Petrus, who raised hand, so he will directly ask you.
Okay.
Hi, thank you very much, Hari, for your presentation. For the last experiments that you showed, the robot was purely simulated, right? Yeah.
That robot is purely simulated.
Okay, and in that case, is the data set that you use of the movement and the entire simulation of the robot available to the public? So if someone, say, decided that they could build a better robot controller, they could try it out on your data set and your platform?
Okay, yes, actually we can make it public. Until now, we didn't get an idea that we could have made it public because the paper is also published. So this is the title of the paper. But I don't think the data is available on the IEEE website. But if you can mail me, I'll basically personally email you the data and simulator also.
Okay, thank you. But I was referring more in if someone, for example, went to your paper and saw, I don't know, some additional materials, repository for your code, that would make it easier for people to one, replicate what you've done, and two, potentially design a build on top of the control system that you've built to make something more or different.
Yeah. So yeah, I mean, but ultimately we were planning to put it on open source, GitHub or something like that. Until now, no, we didn't do it yet.
Okay, thank you.
Thanks a lot. And we have another question from Jens Peterson. Thank you for the talk, Hari. Can you talk about where you are going with this research? Are you planning to connect this to other architectures, functions, or robots? Does this generalize?
Yeah, so for this, I have to say one of the limitations of this approach, because ultimately, if you think strictly application perspective, what cerebral robot is doing is it is considering the errors at every movement step and trying to basically modulate the gains of these errors such that the control performance is improved. But this performance is only specific or local to the trajectory that the robot goes in. And it's not like the cerebellum is learning a model of the robot, that it will be able to generalize to the other simulation also. So here we were actually working with the University of Pavia also, with Alice's team, with Egidio D'Angelo's team, to use the realistic cerebellar spiking network and try to see whether having multiple plasticities at different Purkinje synapses on the cerebellar nuclei would help in consolidating these memories and basically be useful in long term. So as of now, we didn't do it, but we are planning to use these realistic cerebellum simulations to see if different kinds of plasticity will be helpful in forming new motor memories.
Yes, okay, thanks a lot. Yes, very interesting to see how adding details is going to impact. I also have another question actually to both of you, even if your talk were quite on different topics, but I would like to ask you how you had to deal, because probably you had to, with computational load. So from one side, Juan José, all the visualization requires a lot of computational powers. You're using GPUs, but how this is an issue? And for Hari, how is this an issue in view of going to real robot control? So there you need real time, and thus you have the compromise with the computational load.
Mm-hmm.
Okay, do you want to start, Hari, or I? Okay. 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 From my point, in the case of the loads of information, of morphology information, in this case, the application that I have present are designed to work in your desktop, in your personal desktop, you know, then in your personal computer, not in large computers, in clusters, etc. For that, it's not a problem right now, but we are working also with supercomputer version of the applications, and we are dealing with that right now. And it's the thing is that for the moment we have to have the data, the information in the same. If not, we are having problem with network to download all the circuit from another space, because the morphology is it's very large. You know, all the morphology information, it's larger than all the circuit connection between neurons or type of neurons, or of all these more abstract information. The morphology information it's then for the moment, in the case of the version of this application that tries to run in supercomputers, we have to have the information in the same machine. I will say that. I don't know if you have any question and more, something like that.
So yeah, Yeah, for the case of our models, since they are not spiking networks and they are generic neural networks where you can easily perform these, and also the cerebellum doesn't need a back propagation step. The requirement is that the granular layer in the cerebellum produce sufficiently complex signals that can be used linear, like basically that can be used and linearly combined to produce output. And since these are linear operations, we don't need much computational resources. Basically, we also tried to implement the same thing on a robot, and we were able to perform the same learning trials. The problem comes when we try to apply spiking simulations. In that case, I mean, these are like very generic problems then. I think we would need to have specific hardware, such as neuromorphic hardwares, to be able to do it, or we can use the simulators like EdLut, basically from the Granada team. They managed to optimize the computational resources to perform such kind of spiking network simulations. So yeah, we can either use simulators like EdLut, or we have to develop the same code on neuromorphic simulators and be able to also communicate with robot in real time.
Okay, thanks a lot to you both. And it's time to wrap up now, but the questions were not answered, or if you have any other questions, they can be asked by email directly to the speakers, and they will be available and happy to answer. You can already start to save the date for the next HBP Tea Slides session, which will be on the 18th of June, same hour at 2 o'clock. And of course, if you would like to present your own research in the next sessions, send us your application to the email indicated in the slides with the information on your name, university, and the talk you're going to have. And we will be very happy to host your research talk. While all the material, the presentation, and the recordings from this session will be online in the next days. And if you need more information or data or code, you can directly ask to the speaker who are available. And so see you at the next session. And thanks again to the speakers, Juan José and Hari, for their very nice presentations and interaction with all the community. And see you next time then.
Thank you.
Yeah. See you guys.
Bye.
See you.
Bye.
Bye.